Good morning, one and all. Today, my topic is about the cell division and its regulation. Coming on to introduction, cell division is a process by which a parent cell divides into two or more daughter cells. In eukaryotes, there are two distinct types of cell division: vegetative division and reproductive cell division. In vegetative division, each daughter cell is identical to the parent cell, whereas in reproductive cell division, the number of chromosomes in daughter cell is reduced by half to produce haploid gametes. Prokaryotes undergo a vegetative cell division known as binary fission, where the genetic material is segregated equally into two daughter cells. Phases of cell division include interphase, mitosis, meiosis, cytokinesis. The mitosis involves the prophase, metaphase, anaphase, and telophase. This is the cell cycle in which shows the mitotic phase. It is comprises of the prophase, metaphase, anaphase, and telophase. Next, it is a growth and normal metabolic roles. It is G1 of the cell cycle. Then is a DNA replication, which is synthesis phase. And next come to the second growth phase, in which the growth and the preparation for the mitosis takes place. Interphase. Interphase consists of four main stages: G1 phase, S phase, G0 phase, G2 phase. During all these phases, the cell grows by producing proteins and cytoplasmic organelles. G1 is a phase where growth of cell takes place. If the cell does not progress to G1, then the cell enters the stage called G0. S phase, it's also known as synthesis phase, in which the chromosomes are replicated in order for genetic content to be maintained. During G2, the cell undergoes final stages of growth, but it enters the M phase. And this shows the cell cycle. After the cells proceed successfully through the M phase, it may then undergo cell division through cytokinesis. This picture shows the mitosis, in which the mother cell gets replicated and DNA replication takes place. In cell division, two daughter cells are formed. Mitosis. The term mitosis, coined by Walter Fleming in 1882, is derived from the Greek word meaning warp thread. Mitosis is the process of cell duplication or reproduction, during which one cell gives rise to two identically daughter cells. The mitosis used to describe the duplication and distribution of chromosomes, the structures that carry the genetic information. Prophase. Prophase is the first stage of division. During prophase, which occurs after G2 interphase, the cell prepares to divide by tightly condensing its chromosomes and initiating mitotic spindle formation. During interphase, the genetic material in the nucleus consists of loosely packed chromatin. The nuclear envelope is broken down. And in prophase phase, the nuclear envelope is broken down and long strands of chromatin condense to form shorter, more visible strands called chromosomes. The picture shows the centrosome shifting to the opposite poles, and the assembly of the mitotic spindles can be seen, in which the nucleus disappears. The chromosomes with two sister chromatids are seen. Nuclear membrane breaks and it disappears. The nucleus disappears and microtubules attached to the chromosome at kinetochore present in the centromere. Microtubules associated with the alignment and separation of the chromosomes are referred to as spindles and spindle fibers. Next, coming on to the metaphase. Metaphase is a stage in cell division when the chromosomes line up in the middle of the cell by pushing and pulling on centromeres from both chromatids, which causes the chromosomes to move to the center. Anaphase. Anaphase is a very short stage of cell cycle and occurs after the chromosomes align at the mitotic plate. After the chromosomes line up in the middle of cell, spindle fibers will pull them apart. The chromosomes are split apart as the sister chromatids to opposite side of the cell. Telophase is the next phase, the last stage of the cell cycle. At telophase, the polar microtubules continue to lengthen, elongating the cell. The new envelope forms around each set of separated daughter chromosomes and the nucleus disappears. Two nuclear membranes begin to reform and the chromatin begins to unwind. The mitosis is complete. Each daughter cell has identical sets of chromosomes. Next, cytokinesis. First, mitosis ends with the cell nucleus being divided. Cytokinesis begins. During cytokinesis, the cytoplasm of cell is divided in half and the cell membrane grows to enclose each cell, forming two separate cells. The end result of cytokinesis is two genetically identical cells where only one cell existed before. Next, coming on to the meiosis. Meiosis is a special type of cell division which is necessary for sexual reproduction in eukaryotes. Meiosis is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell, as in the production of gametes and plant spores. It produces 4 haploid cells containing 1 copy of each chromosome. Meiosis is the type of cell division by which germ cells are produced. There are 2 meiotic divisions, meiosis 1 and meiosis 2. Daughter cells contain half the number of chromosomes as the original cells. It produces gametes. It occurs in the testes in males, which is known as spermatogenesis, and which occurring in the ovaries in the females, known as oogenesis. During meiosis, DNA replicates once, but the nucleus divides twice. Phases of meiosis. Prophase 1. This includes leptotin, zygotin, pachytin, diplotin, dikinesis, and metaphase 1, anaphase 1, and telophase 1. In prophase 1, DNA is exchanged between the homologous chromosomes in a process called homologous recombination. The paired and replicated chromosomes are called bivalents or tetrads. At this stage, non-sister chromatids may cross over at points called chiasmata. Leptotin, the first stage of prophase leptotin stage, is also known as leptonema from Greek word meaning thread. In this stage, the individual chromosomes begins to condense into long strands within the nucleus. In the picture, we can see a centromere and a centrosome with centrioles also can be seen. Zygotin. The zygotin stage, also known as zygonema from Greek word meaning paired threads. Zygotin occurs as the chromosome approximately line up with each other into homologous chromosomes. The combined homologous chromosomes are said to be bivalent. This shows the bivalent formation, and the formation of synaptonemal complex can also be seen. Next, coming on to the pachytin stage. In pachynemia, the homologous chromosomes becomes much more closely associated. This process is known as synapsis. The synapsed homologous pair of chromosomes is called tetrad because it consists of 4 chromatids. The chromosomes continue to condense. Next, coming on to the diplotin stage. It's also known as diplonema from Greek word meaning 2 threads. During the diplotin stage, the homologous chromosomes separate from one another a little. The chromosomes themselves uncoil a bit, allowing some transcription of DNA. Dikinesis. Chromosomes condense further during the dikinesis stage from Greek word meaning moving through. This is the first point in meiosis where the 4 parts of tetrad is actually visible. In this stage, homologous chromosomes separate further and the chiasmata terminalize, making chiasmata clearly visible. Crossing over. During prophase 1 of the meiosis, a part of chromatid of one chromosome goes over to chromatid of other chromosomes and vice versa. This interchange of the parts of the chromatids of a pair of homologous is known as a crossing over. Crossing over occurs in the first division of meiosis. Importance of crossing over. Crossing over is one of the most important events because it allows variation in produced offsprings. It produces new individuals having new combination of traits. It helps in establishing the concept of linear arrangement of the genes. It also helps in the mapping of the chromosomes and selection of useful recombinations. Next, coming on to metaphase 1. It is the second phase. The tetrads from prophase 1 line up in the middle of the dividing cells rapidly. The spindle fibers get attached to the tetrad from both ends of the cell. Anaphase 1 is the next stage, which begins when the 2 chromosomes of each bivalent separate and start moving from opposite sides of the pole. In anaphase 1, the sister chromatids remain attached at the centromeres and move together towards the poles. In telophase 1, the homologous chromosome pairs reach the pores of the cell. A nuclear envelope forms around each chromosome set. The spindle disappears and cytokinesis follows. Phases of meiosis 2. In meiosis 2, the mitotic division of 2 haploid cells to produce 4 haploid daughter cells. Prophase 2, metaphase 2, anaphase 2, telophase. In prophase 2, the nuclear envelopes break down and spindle apparatus forms. While chromosomes duplicate, place prior to meiosis 1. No new chromosome duplication occurs before meiosis 2. The centrioles duplicate. This occurs by separation of the two members of the pair and then formation of a daughter centriole perpendicular to each original centriole. The two pairs of centrioles separates into centromeres. The nuclear envelope breaks down and the spindle apparatus forms. Coming on to the metaphase 2. The chromosomes become arranged in a metaphase plate. Centromeres are arranged in a line called equatorial plate of invisible spindle apparatus. In anaphase 2, the centromeres separate and two sister chromatids now move towards the opposite poles of the cells. The separated chromatids are now called as chromosomes. In telophase 2, a nuclear envelope forms around each set of chromosomes. Nucleus appears in each nucleus. Chromosomes lengthen and it becomes indistinct. In this picture, you can see the prophase 1, metaphase, anaphase, telophase 1 and 2, and cytokinesis can also be seen. And the importance of meiosis. It produces haploid gametes so that the diploid number of species remains constant generation after generation. Source of genetic variation because crossing over brings together new combination on the chromosomes. Next, coming on to the regulation of cell division. The master controllers of cell cycle are the heterodiametric protein kinases composed of regulatory subunit cyclin and a catalytic subunit cyclin dependent kinase. Two ubiquitin protein ligases and regulatory phosphates. These are the main regulators of the cell division. The bound cyclins regulate the activates of the CDKs. Ubiquitin protein ligases participate in the time destruction of cyclin and other proteins and thereby ensure passage of the cycle is irreversible. In the absence of regulation, cells replicate and divide uncontrollably, leading to disease such as cancer. Cyclically activated protein kinases control cell cycle progression. Cyclical changes in CDK activity are controlled by an array of enzymes and other proteins. There are four classes of cyclins. They are G1 cyclin, G1 or S cyclin, and M cyclin. G1 helps to promote passage to start or the restriction point in late G1. G1A or S cyclins bind CDKs at the end of G1 and commit the cell to DNA replication. S cyclin binds CDKs during S phase and are required for the initiation of DNA replication. M cyclins promote the event of mitosis. The initiation of cell cycle occurs with a receipt of a signal by cell in G0 or G1. The signal induces synthesis of G1 or S phase cyclin CDKs. S phase cyclin CDKs initially are held in check by inhibitors until G1 or S phase cyclin CDKs phosphorylate the inhibitors. This triggers their polyubiquitination by SCF ubiquitin ligase and degradation by proteasome. The released S phase cyclin CDKs then phosphorylate the regulatory proteins, promoting initiation of the DNA synthesis. Mitotic cyclin CDKs phosphorylate large number of proteins that control chromosomes condensation, retraction of nuclear envelope, formation of mitotic spindle, and alignment of chromosomes at the metaphase plate. In this, polyubiquitin is a protein which helps the sister chromatids of the metaphase chromosome to be together. The degradation of this cyclin initiates the anaphase and sister chromatid makes it separate. Due to the loss of mitotic cyclin CDK kinase, activated protein responsible for chromosomal condensation are dephosphorylated. Chromosomes then decondense and nuclear membranes are resynthesized. Cells next move forward into telophase where cytokinesis occurs completing the